Hi. Hi. Welcome to the Stanford MedAI group exchange session. This week, we have Asim Waqas and Aakash Tripathi from Moffitt Cancer Center to talk about their work on building an end-to-end multimodal AI framework for precision oncology. Uh, Asim is an applied research scientist in the machine learning and cancer epidemiology department at Moffitt Cancer Center. Akashi is a machine learning engineer in the machine learning department at Moffitt. They both specialize in multimodal medical AI with a background in the electrical and computer engineering from the University of the South Florida. They work at the intersection of the machine learning and the oncology to improve cancer research and treatment outcomes. They have multiple publications and are working on several projects with high clinical translation impact. Asim and Akashi, thank you very much for joining us today. Before we start, do you have any preference on how you'd like to take questions?
Yeah, we want to keep it as interactive as possible.
Okay, so let's try to make this session as interactive as possible. Without further ado, let's hand it over to our presenters. Thank you.
Thank you, Vijay, for the introduction. Hello, everyone. Can you hear me well? Akash, can you confirm, please?
Yep.
Yes.
Yep.
Great. Thank you. So today we'll present our work, which is a multimodal AI framework for precision oncology, which spans the spectrum from data integration to enabling clinical decision support. So my name is Asim Waqas, and I'm joined by my amazing colleague Aakash Tripathi to present a chain of projects that we have been working on for over the last couple of years. So let's start. So today's talk is something that came into existence rather by necessity and not by choice. This pertains to two researchers with ECE backgrounds trying to understand the complexities of something as diametrically opposite as cancer. And so we will discuss those struggles in chronological order. We started with a review of state of the field and wrote a couple of papers on that. Through those reviews, we identified three issues. One, the absence of co-aligned, co-registered multimodal data for the same patient. Second, the absence of a pre-trained deep learning model for multi-omics molecular data. And last, lack of a multimodal framework capable of processing the full length of multiple modalities that exist in the oncology data. And so we called it MINDS, SeNMo, and HONeYBEE. We started looking at cancer data from the perspective of multiple modalities. And why? Because cancer, just like any other biological system, is multimodal in nature. We gather patients' data in the form of electronic health records, molecular data, radiology images, pathology images, and so on. All of these are individual modalities. And these modalities range in resolution from macro level, such as an organs, site, to micro level, such as tissues, cells, and molecules. Each of these data modalities bring in somewhat different aspect of the disease. So to understand this multimodal problem of cancer, we need to innovate and come up with multimodal solutions. So the hypothesis in our case was that multimodal learning is better than the sum of individual modalities. Now, one way to design this study is to use modality-specific models to get high information content or dense representations of patient data, which is called embeddings from these models. And then once we have these embeddings, we can fuse them together for downstream learning and analysis. But there remains the questions of which modality-specific model is better. Uh, how do we fuse these embeddings? And if at all there is some mutual relational information among these embeddings. And so from the previous slide overlaying the specific components of the pipeline, we can dive into each of these components, and we can actually divide the entire pipeline into three phases. First relates to data development and processing. The second one relates to embedding extraction. And the last one is task-specific multimodal development. So in today's talk, we'll focus on the first two phases, and we'll leave the last component for some subsequent talk. Now, foundation models are these big deep learning models that have been trained on very large data corpus, which is usually beyond the confines of one institution or data entity. In our review, we've seen that these models have consistently shown to perform very well when they are subjected to very large amounts of data. And so we've seen the field where, excuse me, where these foundation models have been adapted for medical and oncology data, and found quite a few interesting works. Some of those we'll include in our talk today. So starting with the first phase of our effort, we started collecting multimodal data, but we realized that there is no single public data source that provides a comprehensive multimodal patient data. Because of this, researchers need to manually scrape these disparate resources to create cohorts that often result in small disjointed data sets. So what this means? This means that researchers like us, we would often end up creating data-specific pipelines. Now, to discuss this part, I'll hand over to Aakash. Aakash.
Thank you, Asim. So, as Asim mentioned, we can't really build multimodal models without having multimodal data from a large enough set of patients. And when we started this project initially, we came across one of the biggest challenges, which still to this day I would argue is not resolved, which is how do you collect publicly accessible data sets of large enough patients with a variety of modalities of information captured with a variety of disease and profiles. And unless there's a large study going on to capture this cohort of patients, it's not really possible. So what we did at that point was we said, okay, let's look at the existing landscape of the oncology data sets and see how we can aggregate and build a multimodal oncology data set using the existing patient information. So to do this, we built a tool called MINDS, which stands for Multimodal Integration of Oncology Data Systems. So can you go to the next slide? Okay, so here's the challenge. Imagine you are a patient within the TCGA study, which is one of the largest multimodal government-led studies that took place. And you as a patient are charged to go to one of these data source collection centers where researchers are going to collect different modalities of data from you. So this could be clinical history, your clinical information, your demographic information, some tissue samples perhaps, and some molecular information. All of this data is linked to you as an individual patient, but to process all of this information, all the different modalities of data is sent to different characterization centers. So, as an example, if your genomic data has gone to genome characterization centers and your imaging has gone to imaging characterization centers. And as a result, when these characterization centers have digitized and anonymized your information, all your information has now been segregated and put into siloed interfaces known as these data commons. Next slide, please. So, as an example, if I were to access that same TCGA patient's multimodal information, I would have to access two separate data common portals. such as the GDC portal, the Genomic Data Commons, where the patient's genomic information, clinical information, and histopathology slides reside. And I would have to access the Imaging Data Commons portal, where that same patient's radiological images, radiological images, as well as a same exact copy of the histological images reside. So to start building a model to get this data, I would have to download and access both the data mechanisms provided by two distinctly different data provider sources, and then manually curate the two disjointed services together myself into a set file structure, and also handle the cases where if a patient's information has been updated for a given study, only one of those institutions may have updated that information. So also handle manually the out of sync information across the different institutions. So to kind of mitigate this manual effort and to scale this up, we said, okay, let's look at the existing tooling that is available to researchers and see if we can map the ontology across the different data commons and harmonize that information and make it accessible through a single API endpoint. And that's what we did. So we accessed the Genomic Data Commons portal, the Proteomic Data Commons portal, the Imaging Data Commons, as well as the Cancer Imaging Archive, and designed those framework in a way that allows for other data sets and data sources to also be onboarded. And through this single unified framework, we are able to programmatically query any patient that exists across this entire cohort and download whatever modality of data is present for those cohort of patients. Originally, we had deployed this in cloud services on AWS and Google GCP, as this is where the raw data itself also resided. So if you were looking to build a query and build a cohort, it was a really fast migration of that data into your, let's say, S3 storage bucket. But realizing that it may not be the most accessible for all researchers and engineers, we have now also made available a locally hostable version of this entire framework and the data warehouse that you can run on a local instance of Postgres. Just as a little bit of background on how we did the mapping and harmonization, we used the Genomic Data Commons data model provided as a foundation on which we built all the other mapping across the different CRDC portals. And from this, we were able to integrate any new patients that are also included live today within the NIH's CRDC. At the time of publication, those numbers were roughly 86,900 patients, so roughly 87,000 patients total within the NIH that we could query. From those 87,000, 41,500 roughly were publicly accessible patients. So we were able to search, we were able to provide all information for those patients directly from our interface for any modality to download. Today, if you wanted to repeat that same process of downloading all the patient's information, you no longer need to go to each one of these data commons providers. You can simply write 4 to 5 lines of Python code to directly query those databases and build a cohort with that query and download every single modality of data present for those patients with only a few lines of code. When that data gets downloaded, along with the data set of all the patient information, you're also provided a manifest file. The goal of this manifest file is to essentially make the data that was requested repeatable. So rather than, for example, if you're doing research today, rather than keeping a copy of the data accessible for other researchers to request from you, for your research to be reproducible, you can simply hold a single manifest file that can be used and shared much more easily to replicate the same exact data set that had been used to train your models. To show this as an example, we are going to download a small cohort of patients' data directly from the GDC and IDC. Here we are just requesting and querying directly the TCGA UAD patients, and we're requesting to download every modality, excluding histopathology slide images. And here you can see it scraped all of GDC and IDC where it found the patients, downloaded their raw data, gave you the manifest file, and for each patient automatically structured every modalities files associated for those patients. And if you now want to share your data set with somebody else, you don't need to share the actual raw data. You just need to share the cohort that you have built. Next slide, please.
All right, thank you, Aakash. So far we have seen the first phase of our project where we were able to develop minds that lets users curate and download multimodal data. Now we look at the second phase, which is to explore the modality-specific embeddings models. In our review of the field, we have explored multiple general purpose as well as modality-specific models, and we came to the conclusion that modality-specific models are far superior than the general purpose models. So here is a list of some of those modality-specific models to name a few. For example, for the clinical data, NVIDIA-sponsored model developed by University of Florida called Catatron has been trained on large amount of EHR data samples. Similarly, PubMed, BERT, and BioClinical BERT are some of the better performing clinical data foundation models. For the pathology data, Virtuo 2 and Uni2H are state of the art till the time we publish this work for whole slide images. For the radiological imaging data, we experimented with multiple models such as RadImageNet, Remedis, Dino versions, etc. But for the molecular data, there was no single model that was capable of processing the multi-omics data spanning across gene expression, protein expression, methylations, mutations. So we developed one in our lab. We call it SeNMo, the self-normalizing multi-omics neural network for pan-cancer prognostication. This was published in International Journal of Molecular Sciences, and the idea was to build a model capable of processing multiple molecular data modalities and use the same model for multiple downstream tasks. So this is the architecture of SeNMo. Once we have the data curated through minds, we use for this process, for this part of the project, we use DNA methylation, gene expression, microRNA, DNA mutation, protein expression, and 4 clinical features, and combine them to train a baseline model called SeNMo, which is a small multi-omics model. And so this was a pre-training on these data across 33 cancer types, covering around more than 11,000 patients from TCGA. So once the model was trained, we used the same model to evaluate on different tasks such as survival prediction, cancer type classification, and some of the nuanced tasks such as TLS ratio, which I'll talk about in a while. Once this baseline model was trained, we used the same model to generate patient embeddings for open access availability. Since this data, molecular data, it suffers from big P, small n problem, wherein the number of features are very high in proportion to the number of samples. So there are very specific, modality-specific pre-processing steps. And so we incorporated within our SeNMo pipeline, we incorporated those pre-processing steps. And once those, each of the omics modality was processed, we then combine all of those modalities to create full-length multi-omic feature vector, which was then used to train our baseline model. So the study design of training, testing, and validation was that we trained a baseline model on data from TCGA, which is publicly available data comprising 33 cancers, and the task was to predict overall survival. Now, once this baseline model was trained, then we use the same model to evaluate on different combinations which are highlighted in red. So for example, for the in-distribution data and in-distribution tasks, we use the same baseline model and fine tune on some of the cancers which were not performing very well once we evaluated the baseline model. The task was kept as same as the overall survival prediction. The next setting was to flip the data and keep the task as in distribution. So from flipping the data means that we use the data which was not used in any form to train the model in training the baseline model. So we used Moffitt's internal lung squamous cell carcinoma data and publicly available CPTAC lung squamous cell carcinoma data to fine tune the model. And the task was in distribution, which was overall survival prediction. The next iteration was to flip the task and keep the data same. So we use the same data, TCGA 33 cancers, fine tune the model to predict a different task, which is primary cancer type. Now, to go further ahead in our simulations, we flipped both the data as well as tasks. So we took Moffitt's internal data, which was not used to train baseline model, and we fine-tuned the baseline model on this data to predict a very nuanced task of tertiary lymphoid structures ratio prediction. So for the baseline pre-training, this is the distribution of the data, the number of cases versus the 33 cancer types. Once the model was trained, we took, before training, we had around 30% of the data held out as test set. So we evaluated the model on that held out test. And so these results are the evaluation of model on that test set. On y-axis are the concordance indices for predicting overall survival. On x-axis are the various cancer types, and any prediction below 0.5 is random. And so you can see that for each cancer type, the model was able to predict overall survival with significant results. For some of the cancers which the model was not able to perform well, we fine-tuned that on that particular data, and then came back to evaluate on held out test set. And these green box plots are either the fine-tuning on underperforming data cohorts or out of distribution data cohort. These are the survival curves to stratify the patients among high, intermediate, and low risk patients for all the cancer types. Now, as I mentioned that we wanted to test this model's capability of undertaking a task which was very nuanced. So the task here was that we selected was to predict the tertiary lymphoid structures ratio in squamous cell carcinoma. And so these TLS are immune aggregates, and they are linked to better survival and responses to immunotherapy. And so TLS ratio is also a validated a prognostic biomarker. So we took 96 patients' data, HND slides stained with CD20. And so those were annotated by expert pathologists to segment the TLS regions. And so we used the multi-omics data, which is RNA expressions and protein expressions, to predict those TLS structures in the data. And so our results show that the baseline model fine-tuned on predicting TLS ratio performed very well and with high concordance with the expert labels, and also were able to stratify with high significance of patient between higher versus low TLS ratios. Aakash?
Thank you, Asim. So once we had developed that one single modality of medical data, where we had a foundation model where we could generate feature vectors, we went back and looked at the data set that we had collected using MINDS and saw surveyed the existing Python frameworks and models to see what models could we use to tackle the embedding generation for the other modalities that we had left. So that included clinical as well as medical imaging, such as histopathology imaging and radiology scans. And we quickly noticed at that moment that there was not a single framework that allowed us to generate embedding vectors in a pipeline that we could use to test different foundation models to see how well they perform in one package. So we started the development of HONeYBEE since we had already done the development of the SeNMo model and the processing for molecular data. We just needed to add clinical and medical imaging to this. And we packaged this all up into HONeYBEE, which is kind of designed to have small workers that go out to these modality-specific data sets and extract only the key features and bring it back to a central database system. Next slide. Okay. So in HONeYBEE currently, we hold 4 different overarching modality processing tools. So one for radiology imaging, one for clinical data, which includes EHR tabular notes, as well as EHR reports and free text data. Molecular, which includes a SeNMo model, as well as the pre-processing required for the SeNMo model, and the histology data. So these are large whole slide images. From all of these models, we generate embedding vectors using pre-trained foundation models or fine-tuned versions of those pre-trained foundation models. And we store all the representative embedding vectors into data sets and make them publicly available on data hosting services like Hugging Face so that researchers can use it for downstream applications. For the clinical data processing, this was an area where we had the least amount of work to do, essentially because Hugging Face has taken care of a lot of the work when it comes to processing textual data to make it compatible with pre-trained language models to generate these features. However, within the medical domain, there are certain things that are more important to increase the quality of the text that you pass into these language models. So, for example, performing feature analysis by using name entity recognition or ontology mapping so that all the documents for all your patients are more harmonized to a single standard can help your final output for whatever downstream application you're using. So what we have done is kind of packaged the most commonly used tools and ontology mapping tools and OCR, for example, if you're dealing with pathology reports, into one application so that you can define the pre-processing pipeline once and apply it across all of your clinical documents and generate embedding vectors for any Hugging Face compatible model, Ollama compatible model, or OpenAI API compatible model. And as an example, here we can take a sample clinical note and we can extract all the entities that are defined by the user or by the existing models that we have onboarded. And from them, you can create opinionated entity schemas so that you can generate a higher quality representation of just the features you're interested in to generate the embedding vectors for using. Next slide. The more interesting one is the pathology images, which is a little bit more complicated to process, simply because these pathology images can be gigapixel large at times, which means essentially they are millions of pixels by millions of pixels. And this could be just for a single sample. And when you're processing these histopathology images, you're not really interested in a single representation of the whole slide. The way these histopathology images are typically processed is by creating tiles of the original histopathology scan. And then for each tile, known as a patch, we generate a feature representation. The issue is that, if you go to the next slide, is that not all the tiles or all the patches are a tissue patch. And you don't necessarily need those samples to generate those embedding vectors for, let's say, the slide on which the tissue resides, or any sort of artifacts such as pen markings made by the pathologist to annotate. If you include those tissue patches for your downstream applications or downstream models, you can end up risking learning those features that do not necessarily correlate at all to the histology of the tissue itself. So out of the box, we include a tissue detector model that takes all the patches in the histopathology scan and automatically classifies each patch as either a tissue patch, an artifact, such as pen marking, or the background slide tissue itself. To our knowledge, currently, most of these libraries that do the histopathology processing use OpenSlide as the backend, which does the processing using the CPU. We have noticed that it is useful and extensible, but it's slow, and it can cause issues if you don't have a high-end CPU core or if you have too many histopathology slides to process, that it can take up too much time and resources. So what we have done is we provide both a CPU-based processor as well as a GPU-based processor using NVIDIA's cuImage library. So if a NVIDIA GPU is detected, it will automatically load the histopathology slides onto the CUDA cores and process them in parallel, giving you a 10 to 20 times speed up for the tissue detection and all subsequent processing. So that's, go back one slide, please. Okay, that's sorry. So once you've detected the tissue, your next step is to generate a higher resolution patches of the areas that actually contain the tissue. So for tissue detection itself, we take a lower resolution scan at a higher level with smaller patches so that we can get a more fine-grained outline of the tissue itself. And then once that tissue region is detected, we create patches from it at a higher resolution scan, but with larger patches, so more histological features are included for each patch. Next slide. All of these tissue patches have variance from slide to slide, as well as institution to institution and scanner to scanner. One way to mitigate this variance is to apply stain normalization across them, and we provide support for the three of the most common methods included, which is Reinhardt, Masenko, and Vadan, as well as, next slide, as well as any sort of stain separation methods you would like to apply to focus on a single stain component. So currently we support H&E stain separation, but we do plan on adding IHC and other stains in the future. Okay, now that you have generated these tissue-only high-resolution patches for all your slides, the next question is, okay, what foundation model will provide the best representation of your original tissue? So for this, we test against three of the most common, popular models that exist for histopathology images. So that's Uni2H, Vicon V2, and Varshai 2. We are able to take each one of these tissue patches and pass it simultaneously to multiple models and generate an embedding vector per tissue patch per model. What you're looking at here are the UMAP dimensionality reduced coloring of those high dimensional embedding vectors down to 3 dimensions, which are then colorized. And it gives you a high-level view to see very quickly which model is able to capture the histological features more accurately. For example, in the Vicon V2, you can see that the histological features are a lot lower resolution. And in the Uni V2, the top and the bottom of the tissue are very similar and are not able to be distinguished, versus in Virchow 2, all 3 regions of the features are distinct and more of the smaller features are better highlighted. Okay. Likewise, for radiology data, we provide a set of standard pre-processing tools as well, which you can develop in a pipeline and apply to your entire radiological set. So in this case, we have spatial standardization and resampling so that in a DICOM volume, for each slice, we can reshape it to fit to the model's input. We provide denoising and artifact reduction, the most standard ones that we can provide for CT scans, MRI scans, and PET scans. And we have a small set of segmentation models supported to, let's say, for example, if you wanted to segment just the lung region in your given DICOM image. And finally, we have a basic intensity normalization so that you can select the Hounsfield's unit within a DICOM image to highlight different regions to your needs. As an example here, we have loaded a CT scan from the TCGA dataset, and we are able to load the DICOM data, load the metadata, store that information, verify all the metadata information, including the DICOM headers as well as the image itself. We're able to select different slices for highlighting which component of the DICOM volume we want to see, as well as window into the correct space so that different parts of the DICOM, different parts of the body are highlighted, like the soft tissue, the bone, or the lung region. Finally, we also, you can see an example of the lung segmentation I was talking about earlier. Here is the molecular pre-processing pipeline that Asim was talking about earlier. This is especially for the SeNMo model that we had developed to take a large set of different multi-omic modalities to generate those feature-rich embedding vectors. Finally, to make this available to other users and researchers to validate this, we have taken several different models for several different modalities and made it available, made all the embedding vectors for that data set available on Hugging Face datasets. To this date, we have amassed over 3,000 downloads, and it has been used quite a bit in a few benchmark studies for models, and we have gone from 17 terabytes of data for the TCGA down to a high rich representation of only 2.5 gigabytes. So we do see potential of scaling, allowing this method to scale on larger data sets to build these models. Finally, all the work is open source and community driven. So all the code, all the documentation and examples of every site you've seen today is publicly available, and we are welcoming contributions so that we can add different pre-processing pipelines for most models that we can support. So on the fly, we can check which model gives us the most unique representation for this specific application a user is looking for.
Aakash, I can take on from here. You can breathe a little bit. So far we have seen that we've generated embeddings from using HONeYBEE for the data that we curated from using MINDS. Now the question is, are these embeddings any good? And for this task, we wanted to evaluate the embeddings extracted from HONeYBEE on different tasks. So the tasks that we selected for evaluating those embeddings were overall survival prediction, classification of patients among the 33 cancer types and sites, and then retrieval of patient based on how close a patient is to another patient in terms of disease. So for this task, we'll go to the results later on, but just to give you the idea of the number of foundation models that we have incorporated inside HONeYBEE for each data modality. So let me take you through those embeddings. So for example, for the clinical data, we have this list of embedding model for clinical data available inside HONeYBEE. And on the right hand side, you can see the 4 UMAP clustering. This is Gatortron embeddings, which is a EHR specific foundation model, then Coin3, which is a embedding models, then MedGemma, which is a medical data specific embeddings model, and then Llama 2, which is a general purpose general LLM. So you see that the quality of the embeddings extracted from each of these models using HONeYBEE they are they're highly predictive of clustering, for example, in this case. Now, to see what type of models are available for pathology data, so we have Uni2, Virtuo, GigaPath, Ficon, MegSlip, which has been recently added, Remitis. And so just with a few lines of code, you can select model and you can feed the data and you'll get the output embeddings for your downstream task. For the radiology data, we've got these types of embedding model specific to radiology data, and similarly with a few lines of code, you can generate embeddings for your radiology images without worrying about the pre-processing steps. As for the molecular data, we use the SeNMo's inference pipeline to generate embeddings for molecular data. So all the pre-processing of the molecular data resides inside SeNMo's framework. Aakash, do you want to talk about some of the results?
Yeah, so if you saw earlier one of these t-SNE plots for the clinical data, and when we mention clinical data, what we mean is essentially those EHR data tables that are describing the patient's demographic information and clinical history information. Something that's more interesting on the clinical side is pathology reports. And if you've ever had to deal with the TCGA pathology reports data, you know that those PDF reports that are handed for this are not always the cleanest. So even with like high-end OCR models, there's a lot of noise present in them, and the information contained within each pathology report doesn't give enough quality information about the patient. So what we wanted to test was, let's say we take medical specific language models and general language models and see if we can fine tune them for our corpus of data and see if the pre-trained version and the fine-tuned version out of that list of total 8 models, which one performs the best and if there's any trends. We had done this for the clinical set, and in the clinical set that we had found was medical specific models were always outperforming. And it was interesting to see that even in the pathology report, where it's a lot more textual data, still the language specific embedding models were outperforming the general language models, even if the general language models were much larger than these embedding only medical models. Next slide. Then we said, okay, well, these embedding vectors are great. These are awesome. But what we're really here for is multimodality. And the reason we're here for multimodality is that not every patient is going to have every single modality present for them, even if they're all within the same disease, same cohort, because not every patient goes through every round of modality collection. Not every person has radiology scans or histopathology scans, irrespective of the disease in this case. So what we need to do is build a model that is modality agnostic so that we can use any and all information present for that given patient to do the prognosis and diagnosis. For this, then we took very basic multimodal fusion techniques like concatenation. So taking the feature vector outputs and concatenating them, or applying a Kronecker product or mean pooling, and to see if those multimodal embedding vectors can compare to the single modalities as they are. And we saw that for the cancer type classification with multimodal fusion, we were able to match the performance of the clinical data, which was the highest quality representation of the patient, which you can imagine. We have direct information of the patient's age, gender, which are highly correlated to their overall outcome. Now, the reason why this multimodal data is important, if you think about it, is that for certain diseases, certain modalities will not be present. And it can be a case where clinical data, which is the most representative, could not be present. And in those cases, these multimodal features can provide a much higher baseline for the overall predictability for your given set of patients. Here, we tried to test that theory. So we took baseline multimodal models that were trained on the BRCA and BLCA cohort of patients. So that's breast cancer, bladder cancer. And these are all multimodal models that were trained from scratch for the overall survival prediction. We took those same cohort patients and then we took the best performing individual modality models and trained with those feature vectors three basic overall survival prediction models. So Cox proportional hazard, random survival forest, and deep surf. And for all three, what we saw is a clear trend, which is if clinical features are present, it is highly predictive of the overall outcome. If they are not present, uh, the multimodal fusion provides, with especially deep serve and concatenation of those embedding vectors, a really, really good and high performance. And in all cases, with multimodality or with individual features and with individual modality, we were able to beat the baseline methods that existed at that point. Just to show the predictive model capabilities of these trained models, here are all the plots for the three survival models, the Cox proportional hazard, the random survival forest, and deep surf, for all individual modalities as well as the simple multimodal fusion modalities for the bladder cancer patients. And you can see for many of them, the stratification is quite strong that we were able to achieve.
Okay, so thank you, Aakash. So let's connect the dots and summarize. So what we have been able to do is we have been able to build a metadata system called Minds, which lets you curate raw data, disparate sources, agnostic of the data type and data sources type, and curate them in a data warehouse where for the same patient, all the modalities data are co-aligned, co-registered in a central location. Once we have this warehouse, we developed a HONeYBEE embeddings framework where we use the individual modalities, picked up modality-specific foundation model, and generated embeddings for each of those modalities. Once we had those modalities, those embeddings, we stored them in an embeddings dataset. We are open source proponents, so we've made all the embeddings available public researchers, available in the Hugging Face, and the code base for HONeYBEE itself is available on GitHub. Now, once we have these embeddings datasets, the researchers are welcome to do any downstream analysis using these embeddings, and we've shown in some of the cases you can do prognosis, diagnosis, and prediction. All the contributions are welcome. We are continuously updating the code base, and if there are any issues, we are addressing them. We are actively managing the repos. And of course, none of this would have been possible without the amazing mentors and colleagues here at Moffitt, and of course, the sponsors for which they have sponsored us through grants. This is our lab page. This is our Hugging Face page. You're open to questions.
Thank you, Asim and Akashi, to introduce this framework to us. Currently, I have a question. Is just when I see you're training your model in the TCGA dataset, there are imbalanced number of the cases for different cancer type. Then how do you deal with the imbalance in the data to maintain a good performance for those like a limited number of cases cancer? Thank you.
Akash, you want to take a shot?
Yeah, I can, and then like you can answer from the perspective of SeNMo training specifically. So the idea here was to like at least on the HONeYBEE side is more to just generate the feature vectors themselves. So you can think about HONeYBEE more as like NumPy library or a PyTorch library. We're just generating the features of a pre-trained model. So these are purely doing inference essentially. But when it comes to training the downstream models on those features, yes, the class imbalance is an issue. And for that, the traditional machine learning applications and traditional, machine learning methods to mitigate that would come into effect. But that would be outside of the scope of HONeYBEE. HONeYBEE would be the feature vector provider, essentially. On that note, I think Asim can answer the SeNMo model, which was one of the models actually trained fully on the TCU data set, and how the class imbalance was handled. He can answer that better.
Yeah, thank you, Akash. So we did you can see the distribution of cancers across distribution of samples across different cancers while training SeNMo. So the class imbalance was a problem, and so we incorporated multiple class balancing techniques which are out there in the literature, such as mode or class weighting, class averaging. So we've incorporated those and mitigated the effect of class imbalance. One aspect that I want to highlight here is that the representation of individual cancers in the test set was balanced. So the evaluation set was balanced. Even though the training data was imbalanced, we wanted to see if what is the model's performance on a test set where each data has equal representations. I hope I've answered your question.
Oh, okay. Thank you. Yes, Amara?
Hi, Asim and Aakash. Thank you very much for the presentation. I joined a little bit late, so my question you might have already answered, but just I'm gonna ask again. The pathology, like it's a framework where pre-trained foundation models can come in and take like this huge dimensional unstructured data, which are the pathology and the radiology and clinical notes, etc., and then create embeddings. I do see that clinical branch in there, which says reports plus EHR. So can we go into the details of like what EHR tables are you including? Billing codes? Are you including labs, vitals? Like what is the EHR component here?
Got it. Yeah. So this can include, honestly, quite literally anything. So the idea here was to not necessarily use models that are designed specifically for EHR systems, like i.e. tabular data only. It's more to adapt tabular data into text and then do the training from there. So essentially, we have key value pairs of any table for any row in a given table, and then you can convert that into text and then adapt it to existing pipelines, if that answers the question.
It almost does, but with given that your focus is cancer, are there tables like which would be like more useful in your opinion? Like because you have worked so extensively on this, so that's why I'm asking.
Billing tables, which will obviously contain the ICD-10 codes, which would be highly representative of the disease that they have. One, social worker notes, surprisingly, is very useful, especially if they are class. That information doesn't really necessarily exist in any of the other modalities or even in tabular data. So if there's social worker notes describing the patient is struggling with financially, transportation, yada, 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 that is highly representative of the patient's outcome. And then finally is pathological notes. So post-surgery notes, as an example, the patient's surgery was a success or not a success, or if there are any complications. Some of those may not necessarily be encoded in any of the other tables that is mostly handwritten out in those pathological notes, as an example.
Any thoughts about labs? Are there any lab values which were, because it's cancer again, like certain labs would have. So that might come in.
Those definitely will. And for the lab values, there's two modes for it, right? So there's one is a physician's notes on the lab value. So it's not necessarily the raw lab values themselves. And that is a highly useful one and would fall in the clinical pipeline. But the full actual lab values that are like in a data table, that would be more on the molecular models side, where we would let one of those models handle it, just because of the density of that. It's not necessarily information that can be captured by a language model, as an example. So it wouldn't be included in this pipeline.
No, perfect. Thank you very much.
Yeah, if I may chime in, the idea was that at every granularity level, we made sure that we are agnostic to data and model. So the framework would be, I mean, you can feed in pathology text report, you can feed in radiology text, you can feed in PDFs. So lab reports, clinical data, EHR, tabular data. So and that this is the reason that in our analysis we've seen that naturally clinical data is the highest carries the highest prognostic value for predicting multiple tasks.
Yes. Thank you.
If you can, sorry, if you
yeah.
could go to slide 6, Asim, I think that'll answer another half of this question, which is that in today's presentation here, what we highlighted were the first two components, which I think, Amara, you may have missed, which is we wanted to see if can we train models not necessarily on the raw data itself, but rather high representation of that data so that we can scale the we can scale rapidly the amount of data and modalities we can use to train these models. And when we're talking about more task specific heads or more specific modalities like pathology reports versus radiology reports being the contributing factor for something, or a specific type of radiology scan or histopathology slide being the contributing factor, that's something that's more on the second half of the screen, which is task specific multimodal model development. Our ultimate goal is to take our framework to generate these embedding vectors at different granularity level of modalities, and then do those analysis on this subset. That answers.
Thank you for your explanation. It's very helpful.
I have a quick follow-up just in this image. For example, there are 4 different modalities, but what do you do if some modality just missing?
Yeah, Asim, do you want to handle this one with the SeNMo?
You could talk about you mentioned this
Through this?
here.
Okay, yeah, perfect. Okay, so our modality combination methods that we have ended up using kind of allows us to handle missing modalities directly. So, for example, if in a given input and concatenation of those patient level feature vectors, if we have a modality completely missing, we can use zero padding for those. If we are using, for example, a transformer model as our downstream model rather than these simplistic models, you can kind of treat every modality's embedding vector as an embedding vector representation of a token. So when you think about all the data input for a patient as these multiple different tokens of different modalities, a transformer model can handle missing modalities directly in that sense because it's not bound by all modalities being present. It's treating each embedding vector as a modality token. So there are ways to handle this. It just depends on how you plan on using the feature vectors you're getting and training the downstream model with it.
So as in Aakash was mentioning that the traditional deep learning models such as an MLP, you don't have much cushion to deal with the missing modalities. All you can do is maximum you can pad, or you can have zero padding or mean padding or max padding. So those are the techniques that you can employ in those. But you see that we mentioned graph neural networks and transformers. For the downstream analysis and task-specific adapters, these are the two families of models which inherently can manage missing modalities without needing to pad zeros or pre-process the missing modalities. So these are a couple of avenues that we are exploring in third stage of our project.
Yeah, thank you for answering this question. Given time limits here, does anyone have last question here? Oh, okay. Okay, thank you, Asim and Aakash today. This is a very good presentation today. Thank you for your sharing your work here. Thank you.
Thank you. And thank you, everybody.
Okay, thank you.